So thank you very much. It's a pleasure being here to talk a little about my work and this new idea, not so new, that we are exploring in our group. It's a pleasure to be with Anil that has been like a reference for my work so many times. Rick and Adri. Adri was my classmate. Great times we had in São Paulo. Ricky, our president of the academy. It's always a honor to be close to you guys. And today I'm gonna share a little bit about the last 9 years of work, maybe a little bit more than that we have been doing in our lab into this thing that started as just a curiosity to find out if we could do research faster. And it grew in type of a different monster that now we are really going all in to find ways to do research without doing research, right? And so the point that I want to highlight very briefly, and I will not give you a lesson in the industry, I'm sure you guys know that so much better than I, is that we use biomaterials everywhere, right? We use biomaterials in, Anil can talk so much about pulp necrosis, vital pulp therapy. You name the condition, there will be a biomaterial there to solve the problem. When we talk about implants, regeneration of the gums, or whatever other situations, surgical treatments, it's very hard to think about a situation in dentistry that a biomaterial will not be involved. And that is so good because that allows us to have a very vibrant community, right? But at the same time, when we think about the way that we do research in biomaterials, it's, I would say in a candid way, is very Jurassic, right? I'm showing here on the screen the findings of my own PhD, on my master degree. And you will see here by the results that I will highlight that since 2005, as I can recount, I was doing research in a very traditional way, right? That means we establish a question, whatever that is. We find out what is our variables, our independent variables. Oh, in this case was the effects of temperatures in properties. So once I want to know the effects of temperature in properties, I select a few temperatures and I measure all the properties for all the temperatures, right? And then I come up with some conclusions, which by the way, were at that time interesting conclusions. But if you look at least 3 out of the 5 experimental groups I studied in my master degree, they all yielded the same results, right? So there was at least a few hours of work that could have been avoided if there were better ways at that time, and maybe the better ways were already there, but were unknown to me, of planning for the research. Because for those that work with ceramics, you know that each specimen takes about like 40 minutes in the oven, plus the cutting, plus the polishing. So when I think about the time that I spent here producing these 3 groups to have exactly the same result, it's something that gives me like something is not right, right? It's a loss of time, it's a loss of resources, a lot of brain power that was just like used for something that didn't yield any good result. So then in 2000, I think around 2014, 15, I came to know about this design of experiments, DOE, which is quite unused in biomedical science and highly used in engineering. And it's not a surprise because in engineering, everything is calculated to reduce time and reduce cost, right? So design of experiments is one field of science, I would say, not really a field of science, but is a portion of the research process. When you think first before start testing, you try to find out which are the things that really matter in an experimental design. And you draw conclusions over time that leads you forward without making you to really test everything that you could test, right? And it's interesting because it allows you to simultaneously testing multiple variables. So instead of testing, like in my case, only the temperature and only the whatever vacuum only the light absorbance, you kind of like test everything at the same time in an organized way. And then you start learning from that and you move the research only with the things that really, really matter. So I'll give you some examples of how radical that is. If you think about, for instance, this case of a study where you're gonna evaluate here. Yeah, I'm back, right? I will share from another computer here. I have it open. Will just be a bit. I'll just stop sharing here. Let me share from this one. Am I sharing the right screen?
Yes.
I am, right? Okay. So if you look at here, if you're gonna test, for instance, material that you're gonna test like 3 properties and you're gonna test 4 different compositions, and every composition you need to print 3 different scaffolds. This is the amount of samples you would need to prepare for this type of study. It's too much. It's just simply too much. It's a lot of time and effort and things that you're gonna be stuck at. And I'm gonna present you one case of our first study in design of experiments that was eye-opening for me. So in this study here, we were actually testing 4 different parameters: pore size, fiber diameter, composition, and number of layers of a 3D printed scaffold. And you can see every one of these parameters were varied 3 times: 50, 100, 150. Then composition, the amount of MTA was 0, 4, and 20%. So there were like several inputs, and every input was already being varied a few times. And when we look at the inputs, then we think about the outputs. What are my outputs? My outputs are my properties. And the properties that we were testing were 5 properties. So if you think about like a full experimental setup for this work, if you only do the matrix there, you're gonna be doing 81 experiments. And if you just do 3 specimens per experiment, you're gonna be producing 243 specimens for this study. And here I'm being very, very candid, because we know that for mechanical properties is at least 10 specimens. It's not even 3 specimens. So by doing the design of experiments, we can actually reduce the entire work from those 80-something groups to only 9. And then we do this allocation in a very pragmatic way. Instead of doing 50-50-01, 50-50-02, we jump from 50 here to 100, from 0 to 4, from 1 to 2, and we do that in a very pragmatic way. And then we just test the properties from only those 9 groups. So you kind of like scatter your data everywhere. And at this point, it's impossible to conclude anything, right? Because you don't know what actually is leading to the variations in the results. But the subsequent steps that you do with statistics, there is a point where you reach something called signal to noise. And the signal to noise is the trick here. because you can find from the signal to noise, for instance, that the composition is affecting at least 4 out of the 5 properties that you studied. And you can see that porosity is mainly, and the porosity is actually the only property that is affected by everything. So that for us is like very interesting, because instead of us studying now pore size, fiber diameter, number of layers, we say, let's just focus on composition. I will never know exactly what is the effect of the others, but I will know the effect of composition, because composition is affecting 4 out of my 5 properties. And then from this, we just go for the biological assessment of our scaffold by varying only the composition of the scaffold. So now you imagine, and I know Anil is very familiar with this type of work. You imagine the time, the cost, and the amount of hours involved if you want to run PCR for 30 days of 81 groups. It's simply impossible to finish. So now that you know that the only thing that really matters in a scaffold is the composition, then I don't mind running PCRs for the groups where composition was the variable. So the point here is that you start understanding the properties in a very broad way, and you start distilling the statistics till you find out which are actually the inputs that will influence my output. So that is what you are trying to find out with one aspect of the design of experiment. What are the things that really matter? The point about in this case, we use something called Taguchi methods, is Taguchi tells you something matters, but you don't know by how much, right? So you can know, for instance, that composition affects 4 properties, but it affects like 2-fold, it affects like 1-fold, it affects like 3 times more than others. It's difficult to assess, but allows you to know this is the input affecting my output. So once we actually know that affected, but we actually don't know by how much, we evolved our work to a second stage where we start using something called meta-heuristic algorithms. So let me give you the case here. Think about calcium silicate cements. And there's a reason why I like studying calcium silicate cements. It's not that I'm in love by the material, but it's because I can change a lot of inputs and get like a material that it will behave more or less the same. But you could do that with resins, but I don't do resins because you need to measure degree of conversion and things like that. So cements is you mix with water, you get the output, right? You get the specimen. So that is why I'm very keen in studying cements for this type of work, where we are actually trying to advance the methodology. So cements are used in many, many cases, right? But they are essentially in the basics of the cement, they are all the same. They are a mixture of powders. And good luck trying to understand that, because you're gonna have like so many different powders, and every powder will contribute towards something, right? So let's see the example. Oh, and by the way, we know the disadvantages, right? I think we are all familiar. Takes a long time, the radiopacity, the variable properties, variable biological properties. So we know that these properties are not the best ones for the materials. And the reason is because it's a mixture of things, right? And this mixture of things make things very complicated for us. Because if you change water powder ratio to increase your flowability, you're going to compromise your mechanical properties. If you think that your mechanical property now is too low and you increase the C3S to increase the mechanical properties, your radiopacity goes bust. And then if you decide to increase your radiopacity by increasing the zirconium oxide, then your mechanical properties go down. So it's a scientific nightmare. to put in a very candid way, because you're going to be trying to adjust mechanical properties by changing the composition, by changing the water powder ratio, but you can never really achieve the best material you would like to have. And this is the first thing we need to acknowledge when we try to develop materials, at least from this methodology, is trade-offs, right? We need to understand that there will be trade-offs, that I will get this and I'm gonna lose that. But what we are trying to find out here is how much of that I'm gonna get and how much of this I'm gonna lose. So then I can have previsibility, right? From the beginning, instead of trying things and learning along the way, I try to have previsibility of what I'm gonna get, what I'm gonna lose. So this work also bothers me for another reason. Regardless of what you have in the market today, it's one size fits all. It doesn't matter if a clinician has a preference. It doesn't matter if the person has open or closed apex. The supplier will provide you MTA white, will provide you MTA repair, whatever brand. And the supplier will say, like, this is to fit this type of patients. And then your preferences and the patient's need are essentially not taken into consideration in this equation. So the question that we had at that time, this work we started around 2018, was which of the components that we should mix and how much water we should use to get the best of these properties. And that time I was naive because I thought we can get the best. No. Along the way, we learn about trade-offs. And I'm going to be discussing the trade-offs with you. So this work is divided in two phases, right? Because the problem is just too big to be answered at once. In phase one, we decided to find out which are the inputs that were leading to variations in the output. Meaning, is the composition, is the radiopacifier, is the amount of water? What is actually really matters when we are trying to solve out this problem of anticipating problems? And once we learn the influence of these inputs in the outputs, then we go to a second stage where we're going to say, okay, now that I know that that matters, I want to understand by how much. I want to understand what is the best that I can achieve for a material. So first phase, we did the same thing. We get like 6 compositions. We had 3 amounts of zirconia and 3 amounts of water. And this gave us a pilot test of 18 groups, right? And you can imagine how happy my students were at that time when I said, okay, we are done with the pilot test, 18 groups, 5 properties. But when we did that, the result was so nice that we actually were able to pinpoint some of the influences that in a way were empirical, but that now we have the data to show that those are the things that really matter. So for instance, why I say it's empirical, because we know that the water powder ratio influences setting time. You can see the more water I have, the higher my setting time. That is empirical. We all know that. And now we can say like, wow, this thing really matters a lot. And then when you go like the water powder ratio and the DTS, that is the diameter tensile strength, we also know the more water, the weaker your cement, but we can see again by the p-value, wow, it matters a lot. And other things we were not so concerned. So if you think like the composition of the powder and the radiopacity, it really doesn't matter. So as long as you care about everything except zirconia, everything except the radiopacifier, it really doesn't matter. You can change your base composition as much as you can. If you're not changing the radiopacifier, your radiopacity should not change. And your composition will not affect the setting time. and your composition will not affect the pH, but there's a trick there we're going to discuss later. And what was interesting for me to see is that the influence of zirconia oxide in radiopacity was really borderline, right? Maybe with more groups that would change. But the thing is that when we were seeing this data in isolation, we were like, actually, the zirconia plays a role in radiopacity. But there's a trade-off here, because if I want to add more zirconia, I need to remove other components of the powder, because I just have 100% powder. I cannot have like 120% powder. So to have more zirconia, I need to decrease the amount of powder. So what do we do with this data here? We rank. And when we rank, you're going to see that this water-powder ratio ranks first in 3 out of the 5 properties, that the powder composition ranks first in 1 property and second in 2 more. And then we say, okay, we already know enough. We already know that powder composition and water-powder ratio matters. Let's move on. Let's by now forget zirconia, because zirconia, we understand, just influences radiopacity. So what do we do with our work after that stage? We study trade-offs. So we get, for instance, water-powder ratio, and instead of testing those 3, we test a lot of them. 1, 2, 3, 4, 5, 6, 7, 8, 9 different water-powder ratios. And then we go for the composition, the base composition of the cement, and we start varying the amount of C3S, because the cement, the base is C3S. And then instead of testing 3, we test quite a lot of them. So what we want to achieve here is something that's called ceteris paribus. The ceteris paribus is everything that happens between an interval. It means I will collect data points, as many as I can, and the statistics, the computer will put all the data points interpolating there, so I can understand really what is going on. And this is very nice to see, because if you look at this data here, you're going to see that if you take the water-powder ratio, there is a certain amount of water that will make your pH to transition from one stage to the other stage. There will be an amount of water that will make your mechanical properties to grow from this amount to that amount. So you know it is around this region. And this data also show us what is in a way is empirical, but now you have the evidence to show that you cannot get the best of the worlds. You cannot get like low flowability and high DTS, because these curves are like C-shape. Either you get one or you're going to get another. You cannot have low setting time and very high flowability. It's empirical, because we know those things depend on water, but now you kind of like start knowing where those things actually happen. And what was nice for us to see in that work is that regardless of the property we were testing, every variation of the property with regards to C3S was linear. So that means that that's great, because now I know this thing is linear. I don't need to test so many. I just can test one here, one there, another there. It's always linear. So as long as you're varying the amount of C3S, don't even bother, because it's a linear regression, right? So with this type of data, we start like really understanding like how these things actually behave. And then we go to the next stage where we say like, oh yeah, very nice. This computer tells us so many nice graphs, curved shapes, straight shapes. Does it really work? And this is where I think a lot of these works on artificial intelligence are failing nowadays, because not everybody is going back to the lab and say, let's see if my prediction really matches the experiments, right? So what we have done here was the next stage, that is the experimental validation. That is like, I get my results, I do the Taguchi, I find out what is relevant. I do my genetic algorithms. I find the curves, and I tell the computer, I want these properties. Tell me how much C3S and how much water I should use. So with those properties, I perform the experiments, and then finally I go for the biological assessment. So what the data has shown us in the red line is the predicted. So is when I go to the computer, say, I want this and this and this and that. I give the five outputs, and I get one amount of C3S. And I go to this other output, and the computer said, use this amount of water. And you can see that, of course, there is experimental variations, but the lines pretty much overlap. Right? Overlap is a bit maybe a strong word, but the lines follow the same trend. So we know that if I want to optimize C3S, that algorithm will tell me how much I need to use for these properties. And if I want to optimize the water content, I can also do the same. And then if we look at the SCM, you're going to see that those cements, and we have a collection of pictures in the paper, they really look like MTA. MTA-like cement, right? C3S, like the needles, you see the crystal growth, and you see like porous structure because of the water loss and things like that, right? A disclaimer here is that we were mixing our powders only with water. There's no additives at all in the liquid, so it's of course not perfect, but still gives us crystal growth, gives us XRD patterns that really look like a cement. So we were achieving nice dental cements there. And then we go for the last stage, where we go for those 5 recipes that the computer told us, oh, these are the properties you're going to get. And then we do cell proliferation. So we find out the cements are biocompatible. And then we do gene expression, we do mineralization, and we find out that the cements are actually bioactive. And that is a discussion I don't want to have if that is bioactive or not. But you can see that the outcome is, the final outcome of the work is still obtaining biological properties. But to reach that with 4 compositions is way different from starting from a point where you're going to test like who knows how many groups, right? So this work we were doing in a very functional way, where one step were leading to the next, and then we understand that property, and then we go to the next and things like that, very pragmatic way. But the outcome is that we were able to prepare silicate cements with predicted properties which were bioactive, right? And though that was a process guided by a computer helping us to decrease the amount of tests that we would be doing. So what we want to achieve in the end is this situation where you're going to have Dr. Kishen with a patient with a periapical condition, and he said, wow, this guy here, he needs higher pH, and you know, I actually needs more radiopacity to follow up this case over time. And in terms of alkalinity or setting time, I don't care very much. This is the composition you're going to use. But if you have another case where alkalinity and radiopacity means a lot, but your setting time needs to be shorter because, you know, it's a patient that, you know, you cannot really keep in the chair for a long time, that will be the composition you're going to use. So it's really tailoring the composition of the cements for the clinician's preference, and also for the needs of the patients. So we went to the 3rd stage of our work, and this is a work that we are doing now since 2020, last year, 24. It's a bit like 1 year and something, and these are unpublished data. But when I was preparing the presentation, I actually think this is really like nice data to really send for publication, and I'm sure that Clarice, who is in the call now, will be very happy to hear that. So this work is being is being championed by my PhD student, Clarice, and with a lot of support from Anna, who is a PhD student that will hopefully join our group soon. And in this work, what we have is the third question. The third question is, we already know how to identify what matters. We know the process of finding how much of the inputs will lead to variations in the output. But we don't have yet a platform that we're just going to be able to predict things without testing. And what I mean about prediction without testing is not that we will not do anything. But you see, when I say DTS or when I say pH is a single measurement, right? We don't know like what is pH over time. We don't know what is the gene expression over time. These things are not like static measures. They change over time. So we want now to move towards a stage in our work that we can do some tests and know exactly what will happen in a month from now. Then we remove this month of work from our lives and we just can do something else, right? So where are we with that work? We are in a new venture called materials informatics. And materials informatics it's kind of like a new field of science, but I would say it's new for us because, again, material scientists, they are using that for quite a long time because they deal with too many crystallographies, they deal with too many monomers and things like that. So what is material informatics is essentially you get like every single material input you can put your hands on, plus experimental data, and you come up with formulas that will allow you to understand if I change this, this is what will happen. And we are trying to add a fourth dimension that is the time, right? So in the case of cements, let's say we're going to get the XRDs, we're going to get the setting times, we're going to get the differentiation potential, we're going to get every single piece of information we have about cements. We're going to put them together and the output will tell us if you have this composition, this will be your differentiation in 1 month from now. Sounds like a dream, but I'm going to share you is the first step and seems to be very exciting. So this is the process, right? So you're going to get the data and then you're going to select what really matters. You're going to train your models. You're going to do virtual screening of your properties compositions, and hopefully you're going to optimize and validate, right? So when we go for where to start, because again, we cannot learn all the properties at the same time. It's impossible, right? And we selected one property that is on the surface seems to be easy, but when we look inside, it's way more complicated. That is pH. And why pH is complicated? Because we have compiled the data from 200 publications, published, I think, in the last 10 or 15 years. So we went there, every single paper, and we start typing and extracting data using some tools we have as well. And we found out, for instance, that if you look at, we always assume that pH and calcium release, these things walk hand in hand because, you know, calcium comes from the calcium hydroxide and it's positively whatever. And then, okay, that should be the higher the calcium, the higher the pH, right? But when we look at the data of 200 papers, actually it's not linear, it's logarithmic. And when we look at the R square, it's pretty bad, right? It's a R square of 0.5. So it means that assumption that we had as researchers that the higher the better, the higher the calcium, the higher the pH, we are not refuting that. We are just saying, you know, it's not that simple. So once that is not that simple, that is what we want to study. So let's get a property which is not simple, and we kind of like have intuition of how that works, and let's develop our model to that. right? Because we cannot start with a property we also don't understand. So this is the first thing we realize. There's no correlation between calcium and ion release. Actually, there is a moderate correlation. It's not linear. pH is such a great property to study. The fact is that when you study pH in papers, and I knew here works in the journals, you know, like people will be measuring pH till the last day of their lives. You start with 3 hours and you go like 12 hours and you go 1 hour and then you're like 30 years old and you're still measuring pH, right? It's a very long property to measure. Again, very easy. You just go there, you put the probe and you get but we are talking about time, right? There's a lot of investment in that. And again, if we can generate a model to simplify that time, probably we can use that model to simplify other times as well. So what we have done was a study on correlations of pH. So what is that? We correlated the pH of these 200 papers from 3 hours to up to 672 hours, that is 28 days. So again, we select all the data, people that reported 3 days, 6 days, 7 days, 28 days. And we see if you know the pH at 3 days, you don't know the pH of 28 days. So you need to measure. And if you know the pH of 3 days, you also don't know much about the pH of 1 week. Keep measuring. 3 hours, sorry. And if you know the pH of 3 hours, maybe you know the pH of 24 hours. Right? So what we see here is that there are weak correlations. That means that if you really want to know the pH of 7 hours, 3 days with certainty, you need to measure for 3 days. There's no other option, right? So what we have done, sorry, this is not loading, but I know my slides by heart. So I'm gonna tell you what is in this slide. In this slide, we have a correlation, 3 machine learning models. So we start from a very simple model and we try to tell the computer, if I give you the pH of 3 days and 3 hours and 24 hours, can you tell me the pH of 3 days, 1 week, and 1 month? Very simple model, terrible outcome. We did a model which was a little bit better. And I said, I give you 3 hours, 1 day. Can you give me 3 days, 7 days, 1 month? Terrible outcome. Then we did another model, and the outcome was so much better. Not ideal, but much, much better. So we learned from these 3 models that if you increase complexity, you start getting closer to truth, but up to a point. Beyond a point, you really need to go and do a model. Right, so this slide here, when you see this, this arrow, is because there is a great increase in R square from the second model, which was moderate complexity, to the third model that was a bit more complex. And when we see this jump, then we know, oh, we can improve this by using algorithms. So then we start doing our own algorithm. And the first thing that we did for that algorithm was what really matters. So if we look at the pH at 3 days, 7 days, and 1 month, what really matters is the pH at 3 and 24 hours and the surface area of your specimen. Everything else, it really doesn't matter, right? But let me do a remark here. When I say it really doesn't matter, it's in the context of our work. I'm not giving you a universal rule here. So when we try to see, we cannot study everything. What really matters here, if I want to predict the pH at 3 days, 1 week, and 28 days, the computer tells me, study the pH at 1 day, study the pH at 3 hours, and study the surface area. Then we move on, and we actually, when we put all those information together, we find out pretty robust model where you can predict the values of 622 hours by just adding surface area, the pH of 3, and the pH of 1 day, 3 hours and 1 day. So if you factor those three functions in your system, you can actually get the pH of 1 month with a pretty good accuracy. And then this part of the graph here, which is very confused, is actually very good for us, because the more confused, the better. It means our model is not like learning from itself. It's really saying, yeah, I learned from the data, and this is my prediction. And by the way, I'm not biased. So you can see that we can predict using data from the literature pretty well what the pH will be. But we need to do validation, right? So the literature tells us we use those data to make our model. Let's go back to the lab and let's see what really happens. So what we did here was we prepare specimens with 3 different surface areas, because we understand surface area matters. We measure the pH of those specimens for 3 hours and 1 day, and we throw in the computer. The computer will tell us, this will be your pH in the end. Then we follow the specimens to see if we were matching what the computer was telling us that would happen. And this was the data. And this was super cool. Because you see, what you see as an X, as an X is predicted. And what you see as a circle is what was measured. This area on the top is commercially available cements, and this is experimental cements. Experimental cements is the same composition of our last paper. So you see, if we get like, I don't know which is this material, let's say it's the Kishan cement, is selling so much in Canada nowadays. If I measure the Kishan cement at 3 hours and 1 day, I get pretty accurate values after 7 to 2 hours. I get pretty accurate at 7 days, and pretty accurate as well at 672, which is 1 month. So we keep following the same cements, and you see that the X and the circle almost overlap. And when we do that for experimental cements, and I like working with experimental cements because then we can do whatever we want. We can just add more zirconia, we can add more C3S. The X's and the circles, they kind of like match again. So it shows that if I just measure 3 hours and 24 hours, we can get very good predictions up to 1 month. And then we went to do some further analysis to find out where are actually our sweet spots here. So one of the things that I want you to look here is these 3 arrows here. What are those 3 arrows? First, they are circles. So you're going to see that the circles are the cements measured at 72 hours. And they are of different colors. Oh, sorry, no, there's a small mistake here. The circles, the spheres, are specimens of 113 millimeters square in area. And you see their R square is always above 0.8. So it means that your chances of having accurate predictions are always above 0.8 when your specimen has 113 millimeters square. Because there is another Achilles heel in research. How big should my specimens be? What is the volume of my specimen compared to the volume of the solution and things like that, right? So we kind of like try to understand, okay, like what is actually the best? The best for us was put 113 millimeters square. You're going to predict with very high certainty what will be your pH after 1 month. And when we do that, when we do the predictions for the commercial materials, you can see that almost every material, we can predict with very high certainty what would be the pH above with a R square above 0.7. And I think 0.7 is a fair, it's a fair R square because commercially available cements are a black box. We don't know how much of each thing we have inside them, right? So you can have, I don't know, like one material that is a pozzolan cement, like the ones we were studying, and one that is a calcium hydroxide base cement, I don't know. So you absolutely don't know what are those things, and you still get a R square of 0.7%, right? So that tells us a lot that the models work pretty well. And here I want also to add something that you can see that these purple boxes here were pretty bad R square. So we're gonna relook at that data because we are unsure if that cement is so wild that doesn't fit the model, or if that is experimental mistake. But we think that is a cement that doesn't fit the model, because we did the specimens all the same way. So even though we have very high, very good R square for 4 out of the 5 commercial cements, there's still room for us to find out if this really fit all the commercial cements. But like I told you, commercial cements are a black box, right? We know more or less, but we don't know beyond a point. And also need to say that commercial cements have also different liquids, not only water. So we don't know how those things could affect. But as long as you are preparing your cements with experimental powders, that could be less of a concern. So what are the conclusions from this third work that we are finishing now? Is that we can kind of like predict the dynamics of pH. This has the potential to cut time and cost quite a lot. So instead of measuring things for 1 month, you just can measure for a day. And this framework, so I see this work as a framework, is if we can do that with pH, we are on track to do that with other properties as well, right? But at this point, we haven't started with other properties. We still are finalizing the studies on the pH. So what are my takeaways from this presentation for you? First, I'm an advocate of design of experiments, right? I think we can learn so much from that. And of course, it requires dedication. It requires much more planning in the beginning. It requires a big dose of statistics. But what I showed you in these 3 pieces of work could never be achieved with traditional methods, right? It would be just like such a humongous amount of work. We couldn't just do that. So what we are looking for, and by the way, I don't apply DOE for all my works, right? Some works still require very traditional approach, but those that we can actually go around the traditional approach, we are trying to go as many times as possible. These approaches undeniably cut cost, cut manpower, and most importantly, simplify data interpretation. And I'll tell you here, there's nothing that scares me more than an interaction of 3 variables. Because if you know that 3 things will interact into an outcome, you don't know anything about your work. I'll give you one example. If color affects the properties and light curing affects the properties and thickness affects the properties, you will never know how properties are being affected by the inputs. So nothing scares me more than have a ANOVA where you put 3 factors and you get like everything interacts. Then I say like, wow, we are just like in big trouble here, because there's no way you're gonna close that conundrum in a lifetime, right? So with DOE, you can actually say like, I don't want to understand the interactions by now. I want to find what matters. And once you find what matters, then you can start studying interactions. And the last takeaway that I would say is like for those interested in using machine learning, AI, whatever other uh, computational tool that you might have access or you're interested about, you need to cater for the validation, right? We are transitioning now from those blue skies research where everything was okay, let's see the potential of AI, to a point where the field is becoming mature very, very fast, right? So if you're going to start a work now that you're going to take like 4 years to complete, and you don't cater for validation in 4 years, the field will be too mature for you to try to publish, right? So try to include in your experimental design of every work you start with computational, the step where you're going to validate your formulas, your algorithms, your interactions, because those days we can see they are coming. It will not take long. And these works take quite a lot of time to be ready. So look at that from the beginning, because you don't want to be caught by surprise when all the work is ready and the outcome will be not very positive because validations might be missing.